Good morning, and thank you all for coming. Because right now we heard a little bit more on the microscopic scale, so I want to bring it back up to an ecosystem scale. It was mentioned several times this morning about ecosystem functions, ecosystem processes, the properties of trees, and following on what Bruce mentioned this morning, it was such a wonderful introduction into the Kauri ecology. So we know within a Kauri stand, it's the dominant tree because of its size, and it stores a massive amount of carbon. And we're all concerned about the concentration of CO2 in the atmosphere and what can be done to capture that and store it. And these massive trees are one of the potential agents. Then also, because of the characteristic of its leaves in the chemistry, we find these considerable buildup of the litter layer, and it comes along with certain chemistry. So the pH is comparatively more acidic to neighboring broadleaf forests. It has the chemistry of the leaves, a high tannin content, which reduces the breakdown. So there is this accumulation of the organic layer with its properties regarding the soil moisture content. And also what Bruce was highlighting, the influence of Kauri on its surrounding plant communities. So if we go or bring it up from an individual tree, which is dominant in its system, but then broadening it to an ecosystem scale, so a Kauri forest stand. So what are the consequences if we lose these fundamental or keystone species? And just to bring us all a little bit back when we're talking about these processes or functions, so what are we actually referring to? So it's all about the interactions between, the living aspects like the Kauri trees, like the understory, and its abiotic factors. And what's the processes within these systems? The main ones are the carbon cycle, which is presented here with the carbon uptake. Then, with the litter fall, we bring it down to the ground. We have a whole suite of fungi and microbial organisms because those ones then involved in breaking it down, releasing the carbon, releasing the nutrients. Then we had mentioned it a little bit, the water cycle. I want to bring your attention to that one as well, especially when we're talking about Kauri stands in the Waitakeres, because it's the main water source of Auckland. So what are the potential impacts of losing these stands on these wider aspects? And then sure, it's all about the nutrient cycling. So we have to think in a larger context when we're assessing these potential impacts. And then sure, another process which is important is the plant and animal succession, which we're observing. So just for example, when we're talking about the carbon cycle, so imagine if we have a breakaway of these massive trees. So will other species then benefit from it? So understory, shrubs, and then may fill a gap with carbon uptake, or have we over long periods of time just not some plant species which is able to capture that amount of CO2 out of the air? With respect, so I've shown the carbon cycle, just also let us remind us about the water cycle. Sure, usually we're not talking about a water shortage, but the opposite has been observed last summer, where we have this considerable, really long period. So what are the consequences then on the whole water cycle, where trees play such an important role? So our specific questions, which we started earlier this year, is to look, what are the impacts on the carbon cycle? And what we were using as indicators were just measuring tree growth, because that's comparatively easy to measure and gives us really a whole lot of information about the productivity and the carbon uptake potential. And litter for the same thing, because it's a really nice indicator of what's cycled within the system. Then with respect to the water cycle, we were just looking into if there is a modification in the canopy, because we know, I mean, these infected trees, they lose their leaves, they lose their branches. So there is not only more light coming through, but there might be more water falling and reaching the ground. And what are the consequences then on the cycling of nutrients, on the cycling of carbon? And then with respect to plant succession, I'd like to refer to a poster which we're presenting, and Donald, who has been working on the project, he is around today as well, so if you have specific questions and potential changes in plant community composition. So where has this all been done? And it's ongoing, and hopefully I'll get another really happy student to continue doing the work, because I just can't do it on my own. So we have been visiting one of those infected sites, and within that stand, with the help of Nick and Lee from Council, we identified trees which still looked comparatively healthy, so only small signs of loss of their leaves, compared to other trees which were within the same stand, which were affected quite considerably. So there were already a lot of leaves lost, so it was definitely more radiation, more sunlight was coming through underneath those trees. And so then we have the so-called dendrometer bands around all the 13 trees which we have been studying, and we are monitoring, and it's comparatively easy to read, so we are monitoring this on a monthly basis, and especially, I mean, the past months have been the most interesting ones, because Kauri picks up its growth rates in spring. So just some of the first results which we are getting. So on the left axis you see the tree growth rates. So this is just presented in millimeter, and it covers the past 2 months. And on the x-axis you have the trees which are only minimally infected, and on the right you see the group of trees which have considerable signs of Kauri dieback. I mean, there is a lot of variability, because this is biology. Each tree is an individual and has its own behavior. So you see large error bars, but definitely a tendency that the healthier looking trees, they're still growing better, they're still taking up CO2 out of the atmosphere. The infected trees, they're not dead yet, they're still growing, but the growth rate is considerably lower. Then also, as another example, or as part of the carbon and nutrient cycle, the litter fall. As you can see on the left, I mean, there are a lot of twigs, brown twigs on the ground. So how to quantify this? So we set up these litter traps underneath, again, the healthy and the strongly affected trees. So we were collecting the litter and bringing it back to the lab. And then shown here is Inge Althusen. She has been on the project for this year, helping. So we are not only just weighed the total mass which we're collecting in the basket, but we separated it, because we wanted to see what's contributing to the loss. Is it the leaves? Is it the twigs? And even more importantly, is it the reproductive material? Because if there are less cones, less seeds produced, those has long-term consequences. So it's really tedious to go through all these different fractions, and then also to do the analytical work with estimating the nutrient content, estimating the carbon content, to see how much has been brought down from the trees. Just one graph, and another graph is shown on the poster here with reproductive material. So what we know from healthy Kauri stands, the reproductive material, it kind of peaks. So most of the seeds and cones come down between February and May. And then, as you can see here, the light gray are the trees which are minimally infected, which still bring down more reproductive material in comparison to the medium and high infected trees, which have a lower proportion of reproductive material. So when this is done this year, I mean, we don't know whether this was, there was a higher shed of the high infected trees last year, or what might be the future. Again, indicating these processes, they're highly variable from year to year. That's why we think it's really, really important to monitor it into the future, especially as we had a really extraordinary summer this year. But again, I mean, this is an observation that we have less reproductive material underneath sick trees, and that has long-term consequences on the recruitment of Kauri. So now to the water cycle. This is Donald setting up one of the so-called rainfall or throughfall collectors. So what we were interested in is to see how much water is passing through the canopy. And our assumption was if there is more gaps, then more water passes through because it's not held up by leaves. And our findings was, so shown is the throughfall, so the amount of water in milliliter we found in the collector you have seen just before. So under the minimal infected trees, there was less water passing through the canopy compared to the trees, or compared to underneath the trees, which were already rather sick. So why is that happening, and what are the potential consequences? So we were using hemispherical photography, so it can be done now with a wide-angle digital camera. As you can already, what's shown here, where there is more canopy openings, it's likely that more water is coming down because it's not intercepted by the leaves in a healthy canopy. I was expecting that also has an impact on the soil moisture content. I haven't seen that. Again, it may be just because it's a super high variability, but also the water coming down may already have been taken up by the understory vegetation. So just to summarize, what we see under the strongly infected trees, they have a lower growth rate, which means less carbon uptake, and if we're talking about the concerns of climate change, that's really important. Amount of reproductive litter is lower, which has long-term consequences into the recruitment of Kauri species, and also what we should monitor closely into the future, and the question earlier this morning indicates what's happening with the water cycle if we see that on a larger scale. Thank you.